Right, this is me, Kinzawi. So today I will be doing a presentation of movement of micromolecules flowchart. Cells synthesize a variety of molecules in order to preserve homeostasis, promote communication. First one is lipid, is a broad class of organic molecules that are hydrophobic and play a central role in energy storage, cell structure. Lipids include fats, oils. The first one is cortisol storage, is a lipid belongs to steroid hormone cells class which is derived from cortisol. A type of lipid, since cortisol is a hydrophobic, can easily pass through cell membranes and bind to intracellular receptors. Cortisol is produced by adrenal cortex, which is within the outer layer of adrenal glands, essential for immunological control, metabolism, and stress hormone. Synthesize and transport a different form, those of molecules based on protein because it is a lipid. Smooth endoplasmic reticulum controls synthesized stars in ER, which are the building block of all steroid hormones. Cholesterol is changed, cortisol synthesized stars. Smooth ER contains enzymes that convert cholesterol into pregnenolone. Then it goes transported into mitochondria. Pregnenolone transported into mitochondria where additional enzymatic modifications occur. Mitochondria contain cytochrome P450 enzymes which help convert pregnenolone into cortisol through multiple steps. Goes to transport vesicles, is a transport in vesicles after it has been produced. Cortisol can be momentarily held in vesicles before to secretion but does not require Golgi alterations. Transferred to Golgi apparatus. Cortisol does not go through a lot of Golgi changes, but it does go through some sorting and packaging. Cortisol is guided toward plasma membrane for secretion by the Golgi apparatus. Secretory vesicles. Vesicles that carry cortisol go through the direction of plasma membrane. Then it goes to the cell membrane as secretion. Cortisol enters bloodstream due to its lipid solubility. Cortisol readily diffuses across cell membrane, attaches itself to target cells. Second one is protein hormone, which is follicle stimulating, composed of chain of amino acids, typically water-soluble and cannot pass through lipid bilayer of cell membrane, so they bind to receptors on the surface of target cells. Is a protein hormone secreted by anterior pituitary gland, play a crucial role in productive females, functions as it promotes the growth of female follicle and production of sperm in males. FSH is produced by ribosomes and travels through the endomembrane system through a difficult and complex pathway. Synthesis and transport pathway. Ribosomes on rough ER. Gene encoding FSH is converted into messenger RNA connected to the rough ER where FSH reproduction starts. Fundamental structure of FSH is formed by ribosomes translating mRNA into polypeptide chain. Rough ER experiences post-translational changes including folding and glycosylation, which is the addition of carbohydrate chains inside the rough ER. Transport vesicles, FSH proteins is inside transport vesicles when it has been folded and changed. Vesicle branches off from rough ER and travels into Golgi apparatus. Golgi apparatus processing. Additional glycosylation takes place in Golgi apparatus to improve the stability and functionality of SHA. For transportation, protein is separated and placed in secretory vesicles. Secretory vesicles, FSH carrying vesicles approach plasma membrane, is released into extracellular environment when the vesicles merge within the plasma membrane. Cell membrane and secretion. In reaction to hormonal cues from hypothalamus, FSH is released into bloodstream through controlled exocytosis. Intracellular protein. Protein synthesized and functions inside the cell. They play a central role in various cellular processes such as signaling and transport and structural support. Actin is a key cytoskeletal protein found in eukaryotic cells. And it is one of the most abundant and highly conserved proteins. Synthesis and transport pathway. Ribosomes in the cytoplasm. Synthesis of actin is carried by free ribosomes instead of rough ER. Ribosome convert into mRNA produced by transcription of the actin gene. Folding and polymerization. Actin is a globular monomer. G-actin after synthesis. Microfilaments are created when G-actin polymerizes into filamentous actin. Intracellular protein. Actin filaments are found in different areas of cell within the cytoplasm. Myosin and other protein facilities the movement of actin filaments to the required location. Incorporation into cytoskeleton, actin filament helps into integrating the cytoskeleton network, which helps in the movement of cells, exocytosis as well as endocytosis trafficking within the cells, moving vesicles and organelles, and cytoskins or cell divisions.